these are your cell respiration notes. This is the final video notes for this unit. So cell respiration is the opposite of photosynthesis. You are taking glucose, C6H12O6, and you're breaking it down. So you're taking all that chemical energy that was stored and you're going to release it, right? We're going to release it into molecules of ATP. We've talked about ATP before. We're finally going to see what this molecule actually is and kind of how it works. ATP stands for adenosine. That's where the A comes from. Tri, meaning 3, phosphate, ATP, right? Remember that every organism needs energy, every organism needs ATP, therefore every organism has to do cell respiration. Even if you are autotrophic, even if you make your own food, right? So plants do cell respiration, bacteria do cell respiration, everything does cell respiration, right? Again, auto means self, troph means feeding, hetero means other, and again, troph means feeding. So animals are heterotrophs, right? Fungi are heterotrophs. There are two types of cell respiration that we're going to talk about. There's aerobic and anaerobic, and we'll go through each of these in turn right now. Let's do aerobic first. So aerobic respiration is breaking down glucose in the presence of oxygen. You need oxygen to do this. And if you look at this formula on the top, what you're going to realize is it's the opposite of photosynthesis, right? The reactants or the substrates here are the products for photosynthesis. The products for respiration here were the reactants for photosynthesis. The only difference being that this energy was sunlight, right? But it's basically the same reaction, just flipped around. This is a pretty lengthy process. It starts in the cytoplasm of a cell, and it's actually going to finish in the mitochondria. So it starts in the cytoplasm and it finishes in the mitochondria. If you are bacteria, well, I'll hold off on that thought. We'll talk about bacteria in a second, right? Here we go. Let's see how this works. We're going to get 36 ATP out of this reaction. Nope, maybe we're not. Maybe I misunderstood my own slides. All right, we're not. We'll go into some detail at the end, but for right now, just know these basic points. It starts in the cytoplasm, ends in the mitochondria, produces about 36 ATP, which is cellular energy. Anaerobic, again, an means without. In this case, aerobic means oxygen, so without oxygen. It's a simpler process. Evolutionarily, it evolved much sooner. It's not as new as aerobic respiration, but you still get some ATP out of it, only 2 to 4, so not nearly as good as the 36 from aerobic respiration, but you still get some. So there are two types of anaerobic respiration, and you do need to know these. The first is alcoholic, and the second is lactic acid. And if you look at the formula, again, here's your substrate, here's glucose, but you're going to notice that there's no oxygen, right? And again, that's because this is an anaerobic process and just doesn't use oxygen. You get carbon dioxide and alcohol, if this is alcoholic fermentation, and 2 ATP, right? Yeast cells are cells that do alcoholic fermentation, right? So if you bake bread, and we'll talk about some examples here in a second, but if you bake bread, the yeast cells that you add to the mixture, if you buy the yeast in a grocery store, give off CO2, which is why bread rises. That's CO2 gas coming out of the yeast. And if you smell it, sometimes people say that alcohol actually smells like bread or vice versa, and that's because the yeast are doing alcoholic fermentation. You're actually making some alcohol. Lactic acid fermentation is done in your muscle cells and just muscle cells of animals in general, but also bacteria, right? So when bacteria break down glucose without oxygen, they're not going to do alcoholic fermentation. They'll do something called lactic acid fermentation. So the formula is a little bit different, but you still get 2 energy out of it, 2 ATP. So let's look at these in a little more detail. Remember, yeast do alcoholic. Again, yeast is a single-celled fungus. Single-celled fungus. When you eat bread that you've made with yeast, you're not eating the yeast cells anymore. They're not alive. They've been destroyed by the high heat. But the bubbles in the bread, right, all these little air bubbles that you see in bread, are the CO2 bubbles that were given off by the yeast as they did alcoholic fermentation, right? And again, these are yeast cells, if you're looking at these. These are single-celled fungi. These little bubbles coming off of them are actually how they reproduce. It's called budding, but we'll talk about that in a different unit. But just if you were curious. There we go. Here's lactic acid fermentation. Remember, these are the 2 types of cells that do it, bacteria and muscle cells. You can use this process to make different types of food. So yogurt's made using bacteria and lactic acid fermentation. Pickles, kimchi, sour cream are all other examples. When you exercise and your muscles are lacking oxygen, so usually when you've been exercising for quite a while, your muscle cells will burn, right? This is why people say feel the burn. They're saying exercise so hard that your cells can't keep up. They use too much oxygen quicker than you can breathe it in. So your muscle cells switch to anaerobic respiration. They switch to this lactic acid fermentation, and this lactic acid builds up in your muscle cells and actually causes them to burn a little bit. That's what the burn is. It's just a buildup of lactic acid. It breaks down pretty quick. It doesn't stay in your cells forever, so your cells don't burn, for minutes and minutes. They usually just burn for a couple seconds. But that's why I'll say it breaks down quickly. And again, it's not as efficient or optimal as aerobic respiration. You're not going to get 36 ATP out of this process, but you do get 2. It's better than nothing. So here's a discussion of ATP. It's not a mysterious mystery molecule. It's adenosine triphosphate. So I want you to think of glucose as being like a $100 bill. It's this big, bulky molecule. It has all of these bonds, these carbon-carbon, carbon-hydrogen bonds in it. And it has a lot of energy, kind of like a $100 bill is worth quite a lot, depending on your view of money. ATP is a much smaller energy molecule. Think of ATP as a more manageable chunk of energy that a cell can use, kind of like a $1 bill. So think about like if you go to Chipotle and you try to buy a burrito or something with a $100 bill. That's overkill, right? You don't need $100 bills to buy a burrito. You only need what? Like somewhere between 5 and 10, right? This is kind of what ATP is like. It's a much more reasonable and much more manageable amount of energy for cells to use. So if you look at adenosine triphosphate and its components, we'll see adenosine when we talk about DNA. If you remember A, T's, G's, and C's from DNA, adenine, thymine, guanine, cytosine. This is the same A as exists in DNA. That's pretty cool. It's just a modified nucleotide is really all it is. There's a sugar in here too, but what I want to draw your attention to is the triphosphate, right? The T and the P, the triphosphate. These phosphates have a negative charge on them, and they want to repel each other. Negative charges will repel other negative charges. But there are enzymes that synthesize ATP, and they force these phosphate groups together. They force them together. And think about this like a spring, like in a car or a shock absorber. If you have a coil or a spring and you're pushing it down, it's going to want to bounce back out. This is kind of what happens when you force these 3 phosphates together. And as a result, you're storing a lot of energy, right? Especially by the time you load this third phosphate into this molecule, there's quite a lot of energy in this bond, right? So here's the first phosphate, the second phosphate, the third phosphate. So the bond in between the second and third phosphates has a lot of energy. That's the bond that enzymes break when cells need to get that energy back. So think of ATP like a spring-loaded molecule that's ready to pop. It's ready to burst at any moment, and it does so by breaking the bond between the second and third phosphate. So because you can recycle ATP once you've broken off that third phosphate, it makes sense to talk about this kind of little loopy diagram here. So let's start at the top. Here's ATP, right? There's adenine, which is the same adenine you'll find in DNA. Here's a ribose, which is the same sugar you find in RNA. That's why it's called ribonucleic acid. Again, this is a modified nucleotide. And here's our 3 phosphate groups. This is ATP. This is a fully loaded energy molecule. You can think about it like a fully charged cell phone that you wake up to in the morning that you've plugged in overnight, and it's nice, fully charged. All right. When you break off that third phosphate, so here we go, the third phosphate is gone. So what you're left with is a D. D means di, adenosine diphosphate, plus the lone phosphate that you ripped off of it, right? So think of this as an uncharged cell phone, right? You've used the energy throughout the day. You still have the phone, but it's uncharged, right? This is what ADP is. It's like an uncharged phone. But you don't throw your phone away at the end of the day. You just plug it back in, right? And cells do the same thing. They reattach this third phosphate back onto ADP to restore ATP again. So it's recycled, right? It's a fully charged phone that you use energy for throughout the day. And then when you're done at the end of the day and the battery's low, you plug it back in and you use it the next morning, right? This is the exact same concept, right? ATP is fully loaded, ready to go. Cells break off that third phosphate to use some energy, and then they have to recharge it. They put the phosphate back on to make ATP again. So from here on out, it's just summary and some new content. So you do need to know the equations. I'm not going to ask you to balance them, but you do need to know them, right? You do need to know the reactants and you do need to know the products, both for aerobic and for anaerobic. All right, so this is nothing new. These are the same equations you've seen in the other slides. I just put them all in the same picture right here. The other thing I would like you to start thinking about is relating photosynthesis to respiration. We talked about how the products of one are the substrates of the other, right? This means they're complementary, right? It means that autotrophs need heterotrophs and vice versa, right? The substrates for photosynthesis are the products for cell respiration. The products for photosynthesis are the substrates for cell respiration. We talked about how, if you're doing cell respiration in this second equation down here, where does this oxygen come from? Right? Where does the oxygen come from? Well, it comes from water, from photosynthesis. Right? Where does the CO2 come from in photosynthesis? Well, a lot of it comes from the CO2 that's given off in cell respiration. So these two equations, these two processes are fundamentally linked to each other. They're fundamentally linked. You can't really separate these two out. They're complementary. Here's a good comparison side by side. Go ahead and take a look at it when you have a minute as part of your notes. I like the comparisons are pretty easy to follow, and we'll review this in class 2. And here's another kind of diagram to show you just the comparison, right? Here's your light energy coming in that's needed for photosynthesis, right? Your organic molecule is your glucose, and oxygen is given off. That glucose is then used in the mitochondria to make ATP, right? And then you give off carbon dioxide and water, which become the substrates for photosynthesis again, right? So these two processes are directly linked to each other. This is the point in the video where I'm going to ask you to stop. Anything beyond this, I will not assess you on, but it's still worth going over just to see a little bit more detail. So here we go. 3 steps in cell respiration. The first is called glycolysis, the second is called the Krebs cycle, and the third is called the ETC, the electron transport chain. So glyco means sugar, glucose, right? Glyco is a prefix to mean sugar, and lysis means to break. So we're going to break sugar. That's not too surprising because we know that's what happens with cell respiration, so it's right there in the name. What you're going to notice is that even though cell respiration as a whole is aerobic, it requires oxygen, certain steps in that process are anaerobic. This particular step, step number 1, glycolysis, is actually anaerobic. You do not need oxygen for this particular step of cell respiration. And what's going to happen is glucose is going to get broken down into what's called pyruvic acid. And it gets you 2 ATP. It gets you a net 2 ATP. You technically make 4, but you actually have to spend 2 of it to make it work. So the net number of ATP is just 2. Right? Keep in mind that the total process, cell respiration as a whole, is going to eventually get you 36. Right? This step only gets you 2, so we still need 34 ATP. So let's hope that our subsequent steps account for this, otherwise our math is going to be really funky here. All right, step 2 is called the Krebs cycle. Krebs is the guy's last name that figured it out. Here's where oxygen starts to play a role. It is aerobic, which means you do need oxygen. And you take the pyruvic acid, it moves into the mitochondria. So far we've been out in the cytoplasm. So here we're out in the mitochondria now. And it gets broken down into water, carbon dioxide, and some ATP. It's actually only 2 ATP again. Right? Again, the water and the CO2 are released. They're not needed. Those are some products of cell respiration. They come from the Krebs cycle. Right? So we've only made 2 ATP. Oops. So at this point, we have 32 more ATP to go. Step 1 and step 2 didn't really get us a lot of ATP. So let's go ahead and keep going. Oh, I forgot there was a formula. Here's the formula for it. You're also going to make these electron carrier buses again. Remember we talked about this in photosynthesis in the bonus info? Here they are again. The molecules are a little bit different, but these are electron buses, and their job is to carry electrons and hydrogens. I'm going to skip over the detail here, but basically when the fully loaded buses, the NADH and the FADH2, arrive at what's called the electron transport chain, they unload all of the electrons, all the hydrogens as well, and that gets you the additional 32 ATP, right? The details are more about AP Bio stuff, but this is the big picture, right? So eventually you do get all 36 ATP. The vast majority come from the electron transport chain. So here is the overall process, and let's walk through it really quick. Right. Here's step one. This is where glucose comes into the cell. The cytosol is the same as the cytoplasm. So we're out in the cytoplasm of the cell. And remember, glucose gets converted into what's called pyruvate or pyruvic acid. Right. This is anaerobic. You don't need oxygen for this. No oxygen. And you only get 2 ATP from this process. All right, that's step number 1. Step number 2 is the Krebs cycle. So pyruvate moves into the mitochondria. So here's our mitochondria. And out of the Krebs cycle, you get an additional 2 ATP, but you also get these electron and hydrogen buses. So here's our little buses, just like we had in photosynthesis, although their names are a little bit different. They're going to go to the electron transport chain, which is also in the mitochondria. And they drop off their cargo, they drop off their electrons and their hydrogen. They return back, they return back to the Krebs cycle once they've dropped their cargo off. There's also some of these buses coming from glycolysis. But here's the electron transport chain, step 3. And the details are unnecessary for you guys, but the overarching picture is that you get 32 ATP out of that process. So a little bit more detail if you wanted it. I hope this helped, and we'll talk more about this in class. See you later.